Yeah, first I'd like to thank C&EN for giving me the chance to be part of the Talented 12 and for everyone who wrote the really nice articles and organizing this really nice symposium. And also I'd like to congratulate everyone else in the Talented 12. As the baby of this group, I am super duper humbled to be amongst all of you. So first, I'm going to give you a little bit of background about me. I was born in Maryland to two Ghanaian American immigrants, and I spent my first 2 years there. At the age of 2, I moved to Ghana, where my parents are from, and lived there for 10 years. That was a great experience. I got to learn about my culture, learn my language. I had the privilege of attending a really great school that really gave me a good background in math sciences. And also really got to essentially gain a lot of interest in certain things that I didn't realize would shape my future, including chemical biology. I then moved to Virginia and attended middle school and high school here in Virginia. And there you could find me either on the tennis court or I also gained a really nice interest in math and science while I, get things marked wrong for spelling it the British way. Math and science is the same everywhere, and that's great. After high school, because of my interest in science, I went to Cornell University, and I often kind of teeter-totter between whether I wanted to be a chemistry major or a biology major. We didn't quite have a chemical biology focus at that time. I ended up being a chem major and took a lot of biology classes, and I think my interest has kind of stayed that path since. Outside of coursework, I actually first joined a lab just for work study. I figured I was a science major and might as well join a lab and see what that's about. I started off in a microbiology lab, first washing dishes, of course, as often many of us do, and then moved on to actually performing some really nice gene mapping in this ciliate here. And then halfway through my undergraduate career, I met Manuel Eriguyen, who was doing some really interesting work with plant biology, specifically medicinal plant biology. I feel like anyone who has like maybe African background always has like a grandma who has some plant that does something. And I thought that was super interesting to bring kind of science to that. So I spent a summer in the Dominican Republic studying medicinal plants, and then came back to Cornell and then studied Saldago canadensis, which is thought to have some anti-diabetic therapeutic potential. After this, I definitely loved research a lot more than I thought I did, and I wasn't entirely sure what to do after my undergraduate career. So I decided to participate in a prep program. So the prep programs are funded by the NIH, and they are specifically for underrepresented minorities in science. And there are a number of institutions. I performed mine at Mount Sinai in New York City. There, I was mentored outside the lab by Terry Cruz, who was an amazing scientist and an amazing mentor, and she was really important for me in helping me kind of craft a graduate school application, which I didn't really have much experience with or have family who really knew anything about. So she was really important to me in that regard. And in terms of research, while I was there, I took a slight turn and did some homology modeling and virtual screening. I didn't have any background at all in any type of computational work. Yet Avner took me on, and he thought it'd be cool for me to also learn about this. So I spent the first 6 months of my year there kind of learning homology modeling, learning the scripts behind that. And then for my last 6 months, Avner kind of put me in a lot of collaborations with different sciences, which one, exposed me to a lot of science, and two, helped me learn how to kind of craft a story for a publication. And it ended up in 2 nice publications with a couple of collaborators, which was great. I then decided to pursue a PhD after my great experience at Mount Sinai. Luckily, I got into a number of programs and decided to choose Yale's pharmacology program due to a number of reasons, including some really cool programs they had, such as the Medical Research Scholars Program, which kind of helped bridge the gap between the bench and medicine, and also because of their really strong chemical biology, labs that they had. Specifically, the work of Craig Cruz. So I actually, for the first time, met Craig during essentially rotation talks where each professor is given 5 minutes to to sell their work to incoming grad students for possible rotation. And when I heard Craig talk, I just could not stop smiling. I thought the work was so cool. And luckily I ended up rotating and joining the lab. So what was so cool about Craig's work? First, I'm going to take a few steps back to just telling you about the life of a protein. So as many of you probably know, DNA is transcribed into RNA, which is translated into proteins, and proteins then go on to perform their intended function. Once a protein performs its intended function, it comes to the end of its life. It has its own half-life. Its death is a little bit more regulated than our death is, in that proteins are tagged with ubiquitin, which leads to their recruitment by the proteasome and to their degradation. So in order to target disease-causing genes, there are lots of different modalities to go after them. To target DNA, there are gene therapies or CRISPR, which is still in the works right now. mRNAs, RNAis are also things that are in development. And at the protein level, there's small molecules and antibodies that are also used. However, what Craig talked about was something that used kind of this last part of the protein's life cycle and looked at using ubiquitination and proteasomal degradation as a possible therapy. So what he calls his tool is a PROTAC, so proteolysis targeting chimeras. And they consist of a protein of interest targeting ligand, a short chemical linker, and an E3 ligase recruiting ligand, which is essentially the degradation machinery that allows for proteins to be ubiquitinated and degraded. And PROTACs work by what we call event-driven pharmacology, where they simply bring together the protein of interest into close proximity with that E3 ligase, which then allows for the protein to be ubiquitinated and degraded. In addition, it's event-driven because it's a catalytic process, and that one small molecule PROTAC can induce the ubiquitination and degradation of more than one protein at a time, one protein molecule at a time. So once I learned about this technology, I wanted to use it for difficult disease-causing proteins. And so I worked on a number of different projects in the lab, but the one that really took off and ended up kind of being my PhD baby was my work on BRAF. So BRAF is a kinase, and what kinases do is that they phosphorylate target proteins, which is another type of post-translational modification. And that leads to a number of downstream effects, such as activation, changes in the localization of a protein, or even changing the different interaction partners of proteins. And these small, more localized effects lead to different things, such as inducing other proteins to be synthesized, changing cell division, motility, growth, or also changing your entire metabolism. So BRAF is specifically a kinase that is mutated in a number of cancers, 8% of all cancers. The most common BRAF mutation is BRAF V600E, so the valine mutated, and essentially causes an upregulation of BRAF's kinase activity, leading to its activation and uncontrolled cell growth, primarily in a number of cancers, including melanoma. So essentially, misregulation of BRAF is an issue. However, due to its prevalence, of V600E specifically, there have been small molecule inhibitors that have been developed. Primarily, the first one developed in 2011 is vemurafenib. And the way it works is by occupancy-driven pharmacology, where it kind of takes the place of its normal substrate ATP and inhibits the protein's function. And small molecule inhibitors are really great therapies, and they've worked really well for oncology. But one issue with them, unfortunately, is that resistance does form to them. And in the case of vemurafenib, this occurs between 6 to 8 months. And mechanisms of resistance include upregulation of the kinase itself, things that completely bypass the kinase and allow for the same function to occur without using that kinase, or additional mutations. And these are acquired resistance mechanisms to BRAF V600E. And beyond that even, so beyond V600E, this 1-point mutation, there, as more cancers and tumors have been sequenced, we've identified several more. So there are class 2 BRAF inhibitors, 2 class 2 BRAF mutants, and class 3, which are really interesting in that they're kinase dead. But all these additional classes have also been shown to drive different cancers. And unfortunately, vemurafenib is intrinsically resistant to both of these. So in general, BRAF is a kinase, and they're inhibitors for one specific mutation, but there really is a need for additional therapies to target this. And this is kind of where our project came in. So in collaboration with a really great chemist in the lab, Saul, we synthesized and designed a number of different PROTACs. I think we ended up designing about 30, and this was our lead at the end, which included vemurafenib as our BRAF recruiting ligand, a short chemical linker, pyrazine-based linker, and then the VHL ligand. So the first thing we did was to evaluate our PROTACs in fibroblast cells that expressed a number of different mutations that have been seen in clinic. And what we saw was that with treatment of our PROTAC, we saw a dose-dependent decrease in BRAF levels, so degradation of the mutant BRAF, which was really great. But even more interesting was that our PROTAC was able to spare wild-type BRAF. So we were able to selectively target only the mutant form of the protein and completely spare the normal functioning protein, which is really great in terms of when you design a drug, because you really want to increase that therapeutic index, and which decreases the chance of toxicity. So we went on to test these in cancer cells and saw similar results and inhibition of the pathway. And those results made us hopeful to test these in a xenograft model in mice. And we found that a 10-day treatment caused a really nice tumor regression in this cell line that is a class 2 BRAF mutant, which is insensitive to vemurafenib, as I said before. So in general here, we're able to take essentially a warhead that is insensitive to one mutant and turn it into a PROTAC, which is able to induce degradation of something that has no therapy right now, which was really wonderful to see. And we did this in collaboration with the Rosen Lab, which were a great set of BRAF biologists to work with. So in summary, why are PROTACs so great and why was I so excited to work with them? One, because of their catalytic mechanism of action, they're able to target proteins that are overexpressed, so you don't have to use more drug to overcome overexpression. In addition, you don't necessarily need something to completely inhibit a protein. You just kind of need a binder to tag your protein in order to recruit them, or in order to induce degradation. And in our project, we're able to show that we're able to selectively do this with acquired and intrinsic resistance mechanisms to BRAF. However, of course, things that completely bypass BRAF are unable to target. In addition, PROTACs are really poised to and degraders in general are poised to expand kind of that druggable space. So right now, therapies unfortunately only touch a small quarter of disease-causing proteins. And due to the mechanism of action of these, we're hopeful that we'll be able to kind of tag these undruggable, quote unquote, proteins and induce degradation. And that's kind of what I'm still excited about and what I'm still working on right now at Monte Rosa Therapeutics, but taking a slightly different path, which is still in the same space. So the way molecular glues work is that they first bind your E3 ligase of interest, and then only in the presence of these are you able to bind your disease-causing protein and induce similar proteasome-mediated degradation. However, molecular glues tend to be smaller drug-like molecules, and for that reason, I thought to maybe be a little bit easier in terms of getting something to the clinic. And in addition, it doesn't need a druggable pocket on your disease-causing protein, which also poises it to kind of to better reach undruggable proteins. So with that, I would like to thank everyone who has been on my journey, from Cornell to the Schlesinger Lab to the prep program, everyone at the Cruz Lab who I interacted with. It was a great learning experience. My collaborators at the Rosen Lab, my thesis committee, and everyone at Monte Rosa, Dom, Sharon, who work with me now. And last but not least, of course, I'd like to thank my family and my partner, Bomboy, for being a huge support system to me and helping me reach where I am today. With that, I'll take any questions.
Thank you so much, Shanique. I was wondering, what happened to your PROTAC? What are the next steps for it, the one you developed?
So unfortunately, I'm no longer in the lab, but I know that I hear that, like, you know, we still have interest in trying a couple of different cancer types, which we didn't necessarily touch on in my project. So that's definitely ongoing right now, in the lab.
Okay, thank you so much.